Greetings, and thank you for joining our webinar. My name is Lorena Benedetti, and I'm a research scientist in the lab of Jennifer Lippico-Schwartz at Janelia Research Campus. On behalf of ASCB and the Public Information Committee, I would like to welcome you to the first of our series of webinars focused on the intersection between art and science. The goal of our webinar series is to illustrate the close relationship between art and science. Art and science ask and address open-ended questions. Both welcome failure as an essential part of the process, and the development of one's aptitude in both disciplines occurs by a continuous feedback loop of thinking and doing. Art and science are based on creativity. Creative work in science and art produces unexpected and surprising results. Surprises stimulate curiosity, which triggers a search to reveal the mystery of things unknown. The development of the microscope enabled the observation of animal and plant tissues as had never been possible, and provide the ability to describe what will later be recognized as the essential unit of a living organism, the cell. With that, I would like to introduce our speaker today. Joe Glavy is an associate professor at University of Texas at Tyler. Joe received his PhD in molecular pharmacology from Albert Einstein College of Medicine and was a postdoctoral fellow under Günter Breuer. His work concentrates on the nucleus and its major transporters, the nuclear pore complexes. Those presentations will focus on the early history of microscopes. If you have any questions, please type them in the Q&A box. And again, thank you everyone for participating. And I'm now happy to turn the attention to Joe.
Okay, Lorena, thank you very much. I'm unmuted, so that's always a good start. Very happy to do this seminar today, this webinar. It's probably the first one of my career, so it's a career moment for me. And it probably was born out of COVID, being the fact we're isolated and we didn't go to meetings, we didn't get to interact too much. And hopefully next year we'll be at a meeting all together and doing some of this live. So today's webinar is in two parts. I'm going to show the collection of microscopes that I've collected through the years, and some of the ones I've bought during COVID. When you're bored in your office, then you might get online and buy some more things. And it really started as not as a hobby, but just a chance meeting of one of these microscopes. And the reason it's part of this seminar series is that early microscopes really become like little statues and pieces of art when you look back on how they're put together and how they're manufactured, the steps, the evolving of the microscope. When I put this title together, I just put a very generic title. And as I went through the talk, the title kept on changing. It was too late for me to change the title, but it is like the evolution of the microscope, the early microscope of the compound microscope. And I concentrate on the French makers because the microscope that wound up on my desk one day several years ago is a French microscope. And I'll turn on the light here so you can see this. And there's a little story. Everything has a little story to it. This microscope was brought to me by one of our admins in the dean's office, my friend Ashley, and she said, could I test it to see if it worked? And she put it on my desk, and I was just in awe of this microscope. I'm in Tyler, Texas, and this antique is put on my desk. And of course, I had my little mag light, which everyone, I think you have a little mag light in your desk. Most scientists do. And I had a slide already on my desk, which, you know, that's what I have. It may be very nerdy, but that's what it's the truth. And it worked. It didn't work super, but it worked very well. And I showed people in the dean's office that it worked, and I left it. I was so tempted to buy it from her, but I had just started here in Tyler, so I didn't want to seem like I was pushy. So about a year or two later, probably a year and a half later, Ashley had moved to the School of Business, and I saw her in the food court, and I asked her how the microscope was doing, and she said it was doing wonderful in her closet at home. So that was an open invitation to make an offer, which I did. And after that, I started to investigate what type of microscope it is. And it just led to a whole new type of hobby, but a collection of of these artworks. And I don't know, in my last lab, which was in New Jersey, there were some old microscopes that were in the benches and everything else. I couldn't throw out a microscope because I felt it was such a craftsman and such a piece of art that we'll find a place for. We'll put it on a shelf, we'll put it somewhere, even if we're not using it. It's really difficult because we know what goes into this, and the time and energy and the craftsmanship is just amazing. So in some ways, the craftsmen start to become the artists and everything. So so this microscope, when I did the research on it, it just led to collecting more of the microscopes that I have. This is a French microscope. Hartneck was the manufacturer of this microscope. What's really telling in this microscope is the double pillar stand. And this was very unique. This is dated back to the 1870s when the kind of the renaissance of the compound microscope and the great age of the microscope development, where it went more from a toy to an actual instrument and biological studies. So the other feature of this microscope, which is very cool, which makes it very unique, it has a round bottom. It has this oval bottom. Very little of the microscopes at that time had this type of oval bottom with a double pillar. So that really was very neat when I was looking through the literature. And a compound microscope in general, and we'll go through this a little bit later, has the objective, and then it has the eyepiece, which you can see here. And the old microscopes, the condensers really were something called a bullseye, which you can see here. And so these bullseyes were a way to focus the light. Not saying it was a good way, but it's the way that the light was focused. And one of the unique features of this is it has, and let's see if I can show this, it has a, the slide holder is one piece. And that really gave away that it was a French microscope. I had some, appraisers look at it and they said it was French, and it's a Hartneck. And there were no patents at that time. So a lot of things were, traded, or they saw something and they made it in their own shops. And it's very unique. So the fact is that this microscope made it to Tyler, Texas, was in a storage locker that Ashley's husband had bought, and then it made to my desk. So that kind of started the amateur collecting of these microscopes, and definitely during COVID, there were more that I added to my collection, okay? And so I have a few more that I'll show to start, and the part one of this, and this is a drum microscope. This is a student drum microscope. It's the simplest design because it's more or less a tube with a cut area where you have the slide will be put, and then you can see here where you have the mirror. Okay, so I have to shine. It's really difficult to visualize this one because it's so small, but you can see the objective, and then you have the eyepiece, and then your mirror. So it's not a high functioning type of microscope. It's a field microscope. And when I was going through the literature, you can see here, there's the ad from 1920. So this is an older microscope, but it's a French microscope that was imported to the US, and a lot of drugstores would have it. And it was a student microscope, a field microscope, where the advantage of this, there weren't too many moving pieces. So it was hard for kids to break this and for students to break it, although they might find a way. But this is dated as the 1920s. This ad is from 1929. And the simple way to focus, you move the tube up and down. So the objective moves. So it has less moving parts. And when I've used this is the easiest one to use because there isn't anything that moves as being an older microscope. Everything is very solid. And so this is a big advantage for a field microscope, that it can be used in the field, and also that it can be so solid. Another student microscope that I then got was a higher model, which has the basic tripod stand, and then it has the mirror down below, which I'll show by the. So you can see that here. You have the mirror, and then again, it has this bullseye condenser. So the pieces that you see if we define a compound microscope, you have two lenses, at least two lenses, and you usually have a condenser for the light. These early models simply have a diaphragm, which is just a rotating diaphragm that controls how much light, just a different size holes that you would see with it, not an actual condenser type of lens to it. And that came on the later models, but these early models didn't have it, and the student models didn't get into that they would have all of these moving pieces. The last one that I'll show in my little demo, take this one out too. This again is around the same time frame. Again, a French microscope. You can see what I call the diaphragm, the rotating diaphragm here. It projects out, and that just controls the amount of light that will come in. Again, you have the same sort of mirror, and you have this bullseye condenser that you see right here. The interesting part of this again is a French microscope, the metal base, the brass apparatus, and this is a tilting pillar microscope. So it can move, and then you can stabilize it by screwing it in, and you can go through different positions with it. And it usually will stay in these positions except today. But this gave a great advantage. You can be straight, or you can be tilted with it and go off to the side, okay? So this French microscope was made by Naichi and his sons, which were one of the famous French microscope makers. And this design was very unique because of this tilting pillar. It was eventually phased out for other types of designs, but it's pretty cool how they put this together. You can see it's got a ball joint for this. And again, this is around 1865, 1870 that this was made. And so at this point, the condenser was the bullseye condenser. And other things that I'll show on the PowerPoint, if the bullseye wasn't present here, you can see it's a lens. They could have a bench condenser, and they used a bench condenser to shine the light through that. Okay, so that's what I'm going to show on my desk cam here. Okay, we're doing very well on time. Another thing before I turn off my desk cam, I want to show you one more thing. That is, these microscopes are in my office, but also I've got a setup to look at these different images I have from EM. So, and I'm just putting this together to show this for the students who think that in the old days, EMs were simple photographs, negative photographs that you would have. Okay, so you can see that pretty well. And I have this in my office. I made a painting out of it, of these EMs that I had through my career in isolating the nuclear envelope. And I just have it in my office because I think all these EMs are so cool. They're so old. This is from about 15 years ago. I did this work with Sam Dales in the Global Lab. But it was one of those moments in my career where I realized the power of these techniques to get to the ultrastructure. And just for the students that might be watching, in the old days, what you would do is you'd have an eyepiece, which I think you can see here, and you go along this. So here's the nuclear envelope, and you can see this. And when we were doing this work, isolating the nuclear envelope, and then when Sam showed this to me and put it on the light box and I was able to look at it, then I realized the power of ultrastructure and the true power of the modern microscope. So I just want to put that as an aside as I go through this talk. Okay, so now I'll switch to my PowerPoint. Switch cameras, don't switch to my screen. There we go. Okay. Just 1 second. There we go. Okay. I can turn my camera to me now. Let's just do that. All right. For me, that's a pretty smooth transition. I don't have my students in class when we're doing this telling me what to do. I've learned finally. So today's talk, first with our demo here, and then with our PowerPoint presentation. At the end, I'll take a poll, and this poll is related to what we're trying to do with the intersection between arts and science through the Public Information Committee, and whether or not we could have these type of displays at our next meetings. I never thought I'd miss meetings so much until now, but when we get to be in person next year, which I'm hopeful for, maybe we can have a booth that would bring some artwork in, some instruments that are classics that we could use as both displays and interactive type of activities. Okay. All right, so I like to do this when I go through a talk, is I have my basic parts. This is like a classroom thing that I've had through Zoom more. Part one has been the collection of microscopes live on my desk cam. We're switching to the PowerPoint. And so now we're going to go through the actual talk. A Google doodle on Angeles Alvaro and her microscope, the early history of microscopes and early designs, the French design microscopes. We'll do a little bit of a recap and with a poll, and then we'll go through question and answer through the QA section of Zoom. So I was going to send some poll questions, but I thought this was a bit personal in this question. When's the last time you used a microscope? When you start to be a PI, sometimes you don't get on the microscope. Students at certain parts, they won't get to work on the microscope at certain parts. I've worked more on the microscope as a PI than I ever did when I was a student or a postdoc. So it's very interesting that this has evolved, and I've done more confocal, I've done more imaging with this. So I'm very grateful for that, because it gives me the opportunity to be interactive and to have my own sets of experiments. So this is your own question. You can say, if it's been too long, which majority of people don't get on as much as they'd like to. The other question here is this basic quote. The most beautiful thing we can experience is the mysterious. It is the source of all true art and science. And just in your head, who do you think said this? Einstein, Newton, Picasso, Barbara McClintock, possible choices. And usually the good quotes are from Einstein. And this is what he said, is that, the mysterious is what we're trying to capture as scientists, and artists are also trying to capture the mysterious and that unknown, and not to be afraid of it, because from the different things we do, we're going to learn. And even with artists, they try to do different things, and they come up with new things that are part of this. And this basic question here, Maria de Los Angeles Alvaro Gonzalez was a cell biologist, histologist, immunologist, marine biologist. Now this question comes out of this Google doodle that was recently put up, and my sister sent this to me, and she said, you should put it in your talk. And then I started to see what she had done in the 50s, and it was quite interesting. The doodle here has her in her microscope. She came to the US in the 50s from Spain, and she discovered 22 different zooplanktons. And this was a major step forward, and she had a Fulbright commission to come to the US. She traveled on ships, and she was able to begin to characterize these different type of zooplankton through her compound microscope. And these are some of the zooplankton that she worked on. And I just thought this was fascinating, all the different work she had done. And I did some more research, and I found a picture of her. And, for this talk, it's really interesting. This was in La Jolla in her office, and it's the intersection of arts and science. So you have a picture of her, of Maria here, and then you have her microscope behind her. And then next to her here is her Picasso. So it is really, the person and the, it says a lot about the person that they have this kind of imagination, and, this would help you in identification of zooplankton. And also there's this caricature of her, which I think is really telling, because it sums around that she was a person, she had her own style and flair, and then she was a scientist. She traveled around the world. She traveled on ships that I don't want to travel on ships myself. And then she discovered this unknown of these different types of zooplankton through her microscope. But really the thing that got me in this is that this Don Quixote print that she had in her office. And in my office, I have my microscopes. And then I have on the other wall, I have the same print, the Don Quixote from Picasso. And my print, there's also the bullfighter and everything else. But I think as scientists, we think of science as a quest, and then we sometimes wonder if it's a mad quest. And that's why I have it in my office, and I don't know if that's why she did, but it was very interesting to learn this and to learn more about different scientists. And there's some more of these doodles that I'll have as we go through this talk here. So back to the microscopes. Okay, so the evolution of the compound microscope and the different components, the inventors of this. And I just kind of put a summation here from the Jansen microscope, the Hook microscope that was made by Thomson. And then we have the von Luenhook microscope, and then on to the drum microscope, and then the varieties that I showed you already, all the way to the compound microscopes. And the thing that I really look at when I look at this is the different types of bases that develop. It's something when you have them and treat them like statues, and how they develop these different types of bases, and then how that affected the function as far as how the mirrors are put together. Then we can see where the condensers are added and different parts of the evolution of the microscope. Okay, so the von Leeuwenhoek microscope was a simple microscope, but it was really a decent microscope, really good microscope, because the lenses were so good. And that's the advantage he had at that time. But it was a simple microscope. And then the compound microscope that Hook and Jansen had used, there were at least 2 lenses. They had a potential for higher magnification. They had condenser lenses in the end. And then, as I said before, the bull's-eye condenser which is a plano convex lens, either could be attached to the microscope or would be a bench type of bullseye condenser that could be shown onto either the mirror or directly onto the sample. So I'm just going to go through the top 3, if you will, of the early microscopes and the start of the cell theory. So father and son, Hans and Zachariah Jansen, made the first compound microscope. That's debated between them and Galileo, but this is the one that was maybe makes the top 1 for this. And it was a first simple compound microscope with 2 lenses. Robert Hooke used a compound microscope to study the first study of the cells, and Antony von Leeuwenhoek used a simple microscope in looking at the different types of bacteria, parasites, protozoans, and it was considered the father of microbiology. Okay, so if we look very quickly at the Jansen microscope, it's a simple tube setup. It's just 3 tubes that come together in a folded type of fashion. All right, and that's very interesting because it allowed for this movement to focus in more on the specimen. So the eyepiece was a bi-convex. The objective was a plano-convex lens. And just to review, these are the different types of lenses. that we have to choose from. This is from Wikipedia, just so you have an idea of this. And you would use, just like a trombone, they would use it to focus. So it was an easy way to focus, but you can see what is lacking. It didn't have any place for the specimen. There had to be another setup in order to see the light source and everything. But it's the first compound microscope back in 1590. Now, Robert Hooke microscope, who was made by John Marshall. I said Thompson before, it was John Marshall. And so this microscope was a compound microscope. And Hooke was a chemist, a mathematician, a pharmacist, and an inventor. So his CV was rather extensive. But his book, Micrographia, which was published in 1665, had 38 plates of different images that he sketched. So these sketchings were either of bugs and different things, but the sketch he has of the core, the bark core, a nice cut of the bark core, was the first time that the cell was described. And in this publication, he described it as rooms in a monastery, because a monastery was a huge type of dorm type of setting. And then what he had used an analogy that this is what you see, and these are the rooms, and he used the term cellula, and that describes as rooms. And that really began where we started to think of, where scientists began to think of the body being made up, or the start of the cell theory, okay? Von Leeuwenhoek, he basically is the first one to describe bacteria. He's called the father of microbiology. And this type of microscope is very simple. But the secret to this, and the secret is that the lenses were much better than anything that had been produced. And he told no one how he made these. And so it kind of went to his deathbed on how he was able to grind out such great lenses for the microscope. So these were Bi-Convex lenses. He made about 25 of these microscopes, and this is very cool. None survived, of course, and anytime you see them, you'll see replicas of these microscopes. And it seems like such a simple design, but because the lens was so good, this is the types of images. And within pond water, he was able to see the movement of these little animals, as they called them. And then from different scrapings, he could see bacteria. So it took the microscope to a different level, even though it's the simplest of microscopes. And again, there's a doodle of him on one of his birthdays, and then they have the Google sign, and then where he can see through the microscope and see the different organisms or the bacterias that he could see in his through his simple microscope. Now, this is actually drawn wrong if you look at it. He's looking through the sample through the lens. It's just the opposite. But so with this, you're able to identify the beginning of microbiology. Okay. And as you go through this, you think of the great scientists of that time and the type of microscopes they used. And the first one I thought of was what type did Darwin use? And he used an aqua microscope, and this was on the Beagle with him. And it's a combination of a simple microscope and a compound microscope. But this was ready to travel. It's more of a field microscope. And this was made by Banks, a British microscope maker. And then in the lab, he would use a compound microscope. And this was made by Smith. These aren't the actual pictures, but these are the type of microscopes that he would use. Louis Pasteur used a French microscope, and it was a simple compound microscope in order to visualize bacteria and yeast. And this one here has got a very solid base. The mirror is attached directly to it. You can see the holders that are present, single holders as opposed to mine, and then the objective, and then the eyepiece that was utilized. So from my collection and doing some research, I really looked at the Parisian manufacturers, the optometrists of this time. And a certain two groups that I really focused on was Naichi and Sons, and then a group that was born that worked in Paris from all over Europe. Oberhausen, Hartneck, and Przemyski were manufacturers that I really enjoyed their microscopes, and I kind of focused on that. So they were German, and Polish that settled in Paris and began to manufacture these microscopes. And I just went through different evolution of their manufacturing and the influences on them. One of them was the drum microscope, like you had seen, and this was made by Benjamin Martin. He was British, and he started this design of the drum microscope. And the advantage with the drum microscope, it's very easy to make, it's very cheap, and it's very reliable. So he began this. He also, so the Martin drum microscope is what he manufactured, and he also manufactured the Culpepper Microscope, which is kind of a derivative of the drum microscope. And this was Edmund Culpepper who first designed this. And then he was Martin was part of the Solar Microscope, which is kind of the predecessor of projectors. And I've never seen one of these, and I would really one day like to see one and see it in action, because it's able to project the image. But I've never seen this. But you know, another influence was Lister, Joseph Jackson Lister, and he solved the problem of and developed the achromatic lens, which changed the diffusion in order to bring the red and the blue lights together in the same focal plane. So you don't have this aberration that you would see that was present in the early microscopes. So these were the two major influencers, which were both British. And if we started on the British manufacturers, we wouldn't end. They have so many of their influences in the development of microscopes. And so Oberhauser, Hartneck, and Przemyski, they worked as a line. So one was the beginning, one was an apprentice, and so on and so forth. And their Martin drum derivative was an inexpensive, stable body form. And then it led to the development, which is the horseshoe footed continental stand, which is really what we're looking at today with the modern types of microscope. So the main business was in Paris, partnered with Hartneck. During the Franco-Persian War, Hartneck had to move back to Germany, and Przemyski took over the business. But these are the ones that I've focused into my collection because it shows really the micro evolution in this, these tradesmen on what they were able to put together and their influences on this. So I can't buy them all, but this is what Hartneck had done. And he had taken the drum microscope and gave it a nice opening with it. So it's basically the same type of stand. And then he opened it so you would have more of the tube body that was exposed, but it was the same premise where you could slide the tube up and down to get your focal, to get in focus. You had a better type of stage for the slides. And this was rather cheap to make and very stable. There's not a lot of moving parts, so that's made this quite profitable and quite inexpensive. But when you look at it, the brass and everything, it's really amazing, the craftsmanship that went into this, the artistry that went into this. They also would take these and have this kind of ghoul type of stand where it would attach basically on the box it was carried in. Its container, you would take it out and then screw it on top, and then you have an even more stable area to work with it, all right? So this was influenced again by the British. They had done this earlier with Carey and Gould, And a lot of manufacturers started doing this, so they have kind of this stable environment that could also be taken out into the field. Which brings me to my microscope, which is a double pillar type of microscope that was manufactured. And so again, it has the double pillar stand, double pillar stand. The mirror is attached to the pillar and the tube body, and then you have your ability to do coarse focusing with it. You can also tilt the microscope by these side panels. You can see that here with the tilting that can be done with these screws here, where the stage and the holder is one piece, and then where we have the ability to do the coarse focusing. Okay, when I take it apart, you can see the objective, you can see the eyepiece, which is here, and then the simple objective. And these are antiques, so you know I'm not buying them for their imaging capabilities. I sent this photograph when I first got this microscope, and I sent it to my friend in Germany and said, I've got a new microscope, and he said, oh, I see that funding is low in the US. So but it's just, I've got it more for the artwork than for how it functions. And what also came with the microscope is some slides. So the student microscopes, I had a bunch of slides that came with it, the ones that survived. You can see here, they're very cool. Foot of fly, root, different things that are there. The one I looked at was called larvae decussant. So this is what I looked at through this double pillar microscope. And again, I'm not, took this picture with an iPhone. I have an iPhone little microscope that helped me a little bit, which is an additive on there. You can see this. I don't know if you can see this, but this is something you can attach to your iPhone. It just clips on and increases your magnification. And I just wanted to show that it works. You can see this as the larvae is right here. I tried to focus on the head of the larvae. And then I did the same thing with the drum microscope. And again, forgive me for not being that good at this, but you can see the head of the larvae here. And then I took it to a modern microscope because I want to see the quality of the slide. And here I have in our lab, we've got a Revolve Microscope, which is idiot-proof. It's very easy to use. It has an iPad and all the modern stuff. And even with this, it's got such a unique design because it's U-shaped and you can flip it. You can have it either as an inverted microscope or an upright microscope. And again, I'm not doing an infomercial, but it's easy. Any student can pop on it and use it. I can always look at quick images. And I looked at this cousin of larvae cousin, and you can see the slide is well preserved. It's fantastic. You can see the head. It almost looks like it's laughing at you. You see the eyes here, the mouth here. It's very cool. So I renamed him Eddie decussant because I think he's, you know, he's an old guy and he still survived very well. So these slides are excellent, and it just shows you how well they were made. And so as we go through the progression with this group, the first stand to stand 2 is the solid base that you can see here. It evolved to the stand 3, which is the horseshoe form, which is the most stable form. And then you can see here that the microscope, the mirror had changed. And so the mirror was detached more and it had more flexibility, which is key because a lot of the drum microscopes, the mirrors were not so flexible. They were in one position and you had to work within the tube. So the step forward is that the microscopes had these lenses that were mirrors that were more adjustable. And you can see down here, the stand 8, the A and the B have this tilting, the A and the B have the tilting type of capability. So you have the stability of the horseshoe base, and then you have the straight on visitation of this, so you can look through. And then now you have the tilt forms, which really, these forms are the predecessor of a lot of the compound microscopes. So lots of groups then switched to not just the French group, different groups started using the horseshoe base. And so let me just show you one example of this. So here we have the Hartnett that is here. And this is the stand 8, just a simple stand. You can see here the condensers there. The mirror is separate, and this doesn't have the tilting capability, but it's an amazing instrument, and that it has this solid base, solid brass, and this is polished up where I don't polish mine. And they would engrave each one of these, and it would fit into a nice kit that you could take anywhere. And the drawer here at is where the slides would go. For 2 of my 4 microscopes that I showed you, 2 of them have this kit, and that's how they, that's where I've gotten these slides from. And so when we look back at Pasteur's microscope, this was a Naichi microscope that he used, very straight on, very similar design. And Naichi and Sons basically became a very famous French microscope maker. They shared designs, and they went into have these binocular and even 3-piece type of microscopes that they developed. And when in my collection, the Naichi is this tilted pillar that we have right here. And so I've covered a lot of the French manufacturers by having these collections of different microscopes. And here, what I was trying to show before that may not showed up, this is the diaphragm, which changes instead of a condenser. This had different size holes, and it controlled the light that way. Not very sophisticated, but the premise of this is really what goes forward into the modern type of microscope. So with this, and I'm at 41 minutes, so I'm doing pretty well. I've only talked about French microscopes because that's what I collect. And even in that microcosm of the whole picture, it's very interesting on what they developed. I just touched on the British, touched a little bit on the German by saying Zeiss right now, and then the Italians and the Americans. There's a lot of different manufacturers, and this, like in the car industry, all these different things that come up, and then what becomes the evolution of the modern type of compound microscope. So what's next? Right now, I'd like to see if you'd like to take a poll, because we're discussing within the Arts and Science working group within the Public Information Committee, should we have a booth? So let's say everything works out and we are able to be in Washington next December and 2022. You know, my idea is that maybe we could put some of this artwork and have, instead of having everything being manufacturers, we could have a booth, and we could have some both displays and type of interactive type of demos, where I think it's very important for students attending the conference that they get the fact that these type of microscopes existed. It's not just an Apple screen, an iPad that you touch and everything comes up. Not that it's good that they struggle, but it's good that they understand the basics of the components that went into it and the history of these microscopes and the art of making the microscopes. And at certain times, like there were certain manufacturers that did their design so they would know it's theirs, and it becomes the craftsmen that go through this. So we have a poll that I think we're going to activate now. Charissa is going to activate the poll that you can take, and we can see what the results. Okay, so the question here is rate your interest in an intersection between art and science booth at the 2022 ASCB annual meeting, right? And so this would be something that would have displays, and then it goes from there as far as what we could do. But if we know you're interested, we can begin to brainstorm and do some more polling of the participants, what they would be interested in this, and seeing different artworks and what we can put together with this, and different types of demos with this. Okay, so again, this is the actual question. If you're extremely interested, very interested, moderately interested, slightly interested, or not at all. And I think if you're not at all, you wouldn't have been attending this. But also the next conference that deals with microscopes, the ASCB, is building simple microscopes for schools. And that'll be on the 28th, which Goldstein will run, and I don't have his first name here. Sorry about that. Dr. Goldstein will run. And I just thought I'd make an advertisement for that, people that are interested in the microscopes and everything. And other things too, while I get the poll results here, kids books. One of the books that I remember as a kid, and this wasn't exactly this one, is these type of scholastic books on the microscope. And I have this one in my collection. It's just great, fantastic. I hope to give it to my grandson one day. It goes through the history in a very simple fashion and can spark the interest of younger students and everything. The other thing too is some of the major books that I have, The Great Age of the Microscope by Turner, and then The History of the Microscope by Clay and Quartz. And these are catalogs where they go through all the different derivatives, some of the styles that made it, some of the styles that didn't. And so this is something I'm glad I have on my shelf to look through this. Okay, so let's just go back here. So far we have 18 participants, and people are extremely interested or very interested. So there's definite interest in this, and I'll take this to the committee to see, you know, if we can do something with this, and then we can then poll members to see what they would have maybe to contribute with this. But again, I have to meet with the committee before that. So we can potentially do this, and I don't know the logistics of it, but I'll make my effort then because everyone seems to be interested in that. So what I want to do at this point, I want to thank everyone for taking the time. I know we're all busy. We have a million things going on. The real reason that I've done this is to share and to go through the different things that I have done and, you know, make some competition in this field of amateur collecting. But I'm glad I have these microscopes because it inspires me, and that's, I think, what the artwork is supposed to do. They're works of art at this point, not super microscopes anymore. They're past their prime, but it's a conversation piece, especially with students, and gets you stimulated about what you're doing. We should know both the history and the art of making a microscope and everything else. So I want to thank you, and now we have time for questions. Okay.
Thank you so much, Joe. It was a very terrific presentation. Thank you so much.
Oh, thank you.
Can you see, we already got some questions from the audience. So first question is, have the pieces in your collection required any restoration, and do you do the restoration by yourself?
I haven't done any restoration. That's a bit high end for me because I like the way they look right as they are. I've tried to clean the lenses as much as possible, but I like the rustic look of them. So I haven't done anything with that because they're pieces of art to me, and their age is part of their art and everything. I just find that to be very interesting. They held this up this well over these times and everything. So I'm not interested in how shiny they are. I'm interested in more authentic they would be, and not trying to take off a little bit of, you know, the wear and tear of their age.
We have another interesting question that is, how did the light sources evolve prior to our modern laser or LED options?
That's a great question. The light microscope, the mirror and the condenser really help focus the products, focus the light onto the specimen. Until you turned on a light, the light microscope was a simple mirror. And what I talked about, the flexibility of the mirror, and then the diaphragm diameter and the light source or the lens source going through it helped really focus the samples. So in what I've talked about, we're getting up to really where they started to add the condensers and then concentrate the light coming in. When I talked about the Martin Solar Microscope, that's something I think is very interesting because it was intended to be a microscope and became more of a projector. So one of my goals is to find out more about this. And one of my goals really is to go to these different museums that exist and see if I can see these microscopes and maybe see how they work, the ones I don't have and the ones I can't afford. But really, I think the light source is the next step on concentrating on that, and the condensers really did that. And until electricity was really, utilized, you had to rely on sunlight. With the solar microscope, what I've read is they put it, out on a windowsill, when the light when the sun was brightest, and went into a dark room or a dark area in order to visualize the specimen. So I think that's super cool. I'd love to look at one of those and see how they work, because they're really trying to concentrate on the light source and concentrate the light source from there. And what can I tell you about the next question is about the about electron microscopes, and that's a great question. I think that could be in some form another webinar where we talk more about that. I've done work with collaborators on cryo-EM, and they're more the experts on EM. I'm more the fan on it, and able to visualize this. And I have to tell you, the reason I showed you the EMs that I have is just because that was a point in my career where I said, okay, this is what I'm going to do. I'm going to get down to the ultrastructure. And it just took a simple, negative print, a negative to show me this, and able to give my career a better direction. And we went on to look at the nuclear pore complex at the ultrastructure level and stuff. The ins and outs of electron microscopes is probably for another lecture and a better speaker.
I have a question related to the one before. So at the beginning, it seems that the microscope was engineered, built, and developed by the scientist itself. And looking at these very early microscopes, they look pretty expensive in their engineering and their building. So who were they accessible to? Who could afford this type of instruments and can use them?
Well, for Luenhook, he was a successful businessman, and he was a clothing manufacturer. So he was trying to look at different threads and focus on the threads more than anything. So it came out of his business where he developed this microscope, and first the lenses and then a lens holder, then became a microscope. Hook had done other things, so he was able to afford the microscopes that were manufactured for his studies. So yes, it became kind of an aristocratic type of hobby that led to, or the optometrists themselves that were able to afford these microscopes and manufacture them.
Joe, there are another couple of questions here. One is, are spectroscopy and microscopy intertwined?
Okay, so how are they intertwined?
Or rather, are they?
Well, of course they're intertwined. I don't know what they mean by that question.
Yeah, that's a good question. We would need to get some clarification on that.
Yeah. And okay, so I've got what I also want to add is I have a list of the references, because Brian Stevenson is on, who's watched the talk, and he's written a nice paper on the French microscopes. I didn't upload my Word file where I've got the whole list of references as far as what to, there's also a video on Van Leeuwenhoek that I think I can, maybe I still can do that, that I could put in the chat so everyone has the proper references for my talk.
That's great, Joe. I was thinking, as you were giving your presentation, that of course, like in the simple microscopes for color illumination, the light source was visible, in the focal plane of the specimen. So it would be interesting to possibly have sort of a second webinar in this series that was focused on new innovations in microscopy that introduced other elements into the optical path.
Yes, I think that would be good. And I feel from our committee, the reason to start this way is so we can do more and have some ideas for further seminars on this.
Absolutely.
And the imaging is very, very important. And, the resolution and the clarity became more and more where we can see, with better resolution of the specimens and the cells.
Right. Lorena, do you have any closing remarks?
Well, I would like to thank again Joe for the very beautiful presentation and the work that he did. And I think that the ASCB should be really excited to have people like you that have such interest and passion and have the time to collect these pieces of art and preserve them. So that's really great. And I want to thank everyone for joining us today. And we hope you could join all the webinars we prepared that are coming up from ASCB. Thank you, everyone, for joining us.